Two.
Hello, everyone, and welcome to today's DDN webinar. I'm Tiffany Garbett, Assistant Science Editor for DDN, and I'll be moderating our discussion. We have an exciting webinar planned for you today. Our speakers, Dr. Matthew Disney and Dr. Stephen Thomas, will discuss therapeutic solutions to COVID-19 and delve into the highs and lows of developing pandemic drugs and vaccines at lightning speed. After the talk, Dr. Disney and Dr. Thomas will participate in a live Q&A session. To submit your questions or comments, simply submit your question to the Q&A portal to the right of your screen. We will try to get to as many of these as possible. I'd like to take this opportunity to thank our webinar sponsors, ACD Bio, Taconic Biosciences, Cayman Chemical, and Olink. ACD Bio. ACD, a biotechny brand, shortens the path to personalized medicine and enables research, drug development, and clinical applications by unlocking the power of RNA. ACD is a leader in molecular pathology, developing cell and tissue-based research and diagnostic tests for personalized medicine. ACD's products and pharma assay services are based on its proprietary RNAscope technology, the first multiplex fluorescent chromogenic in situ hybridization platform capable of detecting and quantifying single molecules of RNA in situ. ACD's RNAscope, BaseScope, and pharma assay services allow customers to run assays for their unique targets rapidly. Taconic Biosciences. Taconic Biosciences is a fully licensed global leader in genetically engineered rodent models and services. Founded in 1952, Taconic provides the best animal solutions so that customers can acquire, custom generate, breed, precondition, test, and distribute valuable research models worldwide. Taconic uniquely combines this broad portfolio with expertise in key areas such as microbiome, immuno-oncology, metabolic disease, and more. Cayman Chemical. Cayman Chemical draws from more than 40 years of research and manufacturing experience to develop tools to support coronavirus research. This includes anti-coronavirus compounds and screening libraries for drug discovery, as well as serological SARS-CoV-2 ELISAs and neutralizing antibody controls to evaluate the immune response. Cayman also offers in silico modeling contract services that can predict the binding of investigational therapeutics to SARS-CoV-2 targets. Olink. Olink offers an unmatched high multiplex technique to identify actionable biomarkers with a strong focus on the human plasma proteome. Using minimal sample volume, Olink provides high-throughput, quantifiable results with exceptional sensitivity and specificity and a dynamic coverage range. Olink's mission is to accelerate proteomics with the scientific community across multiple disease areas to enable new discoveries and better understand complex real-time human biology. Olink works with a growing number of core labs around the world, including Europe, the USA, and Asia, offering analysis and support to an expanding global customer base. Our sponsors have provided us with some helpful handouts related to today's webinar. You can find and access these in our handout section located on the right side of your screen. And with that, let me introduce our first speaker, Dr. Matthew Disney. Dr. Disney earned his PhD in biophysical chemistry from the University of Rochester in 2003 and completed a postdoctoral fellowship in organic chemistry at the Massachusetts Institute of Technology. In 2005, he joined the Department of Chemistry at the University of Buffalo as an assistant professor and later joined the Scripps Research Institute Florida campus in 2010. Dr. Disney's research is centered on understanding the molecular recognition of RNA by small molecules. His team develops novel experimental approaches that provide a foundational understanding of RNA small molecule interactions. In 2003, his team was awarded the Eli Lilly Award in Biological Chemistry from the Biological Chemistry Division of the American Chemical Society. Dr. Disney's team also develops and implements computational tools to design bioactive small molecules that target RNAs across the transcriptome. They have identified multiple small molecules that target microRNAs involved in cancer, addiction, and viral infections. Dr. Disney also serves as an associate editor for the Journal of the American Chemical Society. His research provides sequence-based drug design across the human transcriptome to provide precision-led medicines. Dr. Disney, let's just take a minute to make sure your slides are up and running. And it looks like they are. Dr. Disney, please feel free to begin. Thank you.
How my group thinks about
targeting RNA with small molecules broadly. And then I'm going to describe applying these tools across the entire SARS-CoV-2 genome. I think many of you are sensitive to this, that I think we're in an amazing time to do science. And there's a few predictions that I would like to make that will come true, is that it will be more amazing to do science next year and the year after that and the year after that. One of the ways in which science has been revolutionized, especially in biomedical science, is sequencing the human genome. And so in 2001, this was the first draft of the human genome, was disclosed. Eric Lander, who's now a cabinet member position in the Biden-Harris administration, representing all of us scientists. Francis Collins, who's the director of NIH, led the public consortium that was the team that published that genome. Craig Ventnor did shotgun sequencing with Celera Genomics. We've seen a huge amount of efficiency in next generation sequencing, and part of this is due to deploying and implementing new technologies. And one thing that we need to be sensitive, I think we take this for granted, well, previous to this year anyway, we did, is that just simply the efficiency by which we're able to conduct experiments now far outstrips what people could have imagined a long time ago. So one of the examples of this is this orange line is Moore's law. So it's the constant doubling of computational power that occurs versus the cost of a genome, which is in blue. And you can see that the increase in efficiency in genome sequencing is far outstripped computational power. And the consequence of that is, in terms of computational power, is computation that got a man on the moon 51 years ago. We all have 100,000 times that computational power that's sitting on our wrist if we have a smartwatch, or sitting in our pockets if we have a smartphone. And if you don't have one of those, you're stuck in the past, I think. One of the things that was most interesting about that, and it's going to come into view in SARS-CoV-2, is that RNA was found very early on to play a tremendously important role in not only the human genome, but other genomes like viral genomes. And so, for example, the amount of protein that's made from the human genome is represented here by this orange dot to the left. It's about 1% of our genome encodes protein, yet 80% of that genome codes RNA. Now, the target that we're going to describe here is SARS-CoV-2, and that its entire genome is made of RNA. And one thing that we need to consider in terms of druggability, which is part of what we're trying to focus on here, is that when we think about drugging RNA with small molecules, basically 0% of them have been targeted. The most common way to target an RNA is using antisense oligonucleotides that read out sequence. As I'll show in this talk, and what we well know now, is that a lot of these functions of RNA in the human viral genomes occurs because of regions of the RNA that don't necessarily encode for protein, but certainly control and regulate the amount of protein that's made from particular messenger RNA or transcript. And so what my group has been doing for the past 15 years is we thought that developing small molecule modalities that target RNA would be very important in bioscience generally, not only to deliver precision medicines for a variety of indications for which we have no known treatments, like ALS, for example, muscular dystrophy, also to deliver lead medicines against emerging pathogens. And so I'm just going to talk about our group has done a lot of work on this. And so we've, for example, we've done library versus library screens, because prior to our work, there wasn't a lot of information known about what are the RNA folds that bind small molecules, or the types of small molecules that bind RNA folds. We have a library versus library screen to very quickly identify RNA folds that bind drugs and drugs that bind RNA folds. We've been able to integrate that information into RNA structure prediction algorithms, where literally we can take a genome sequence or an RNA sequence, SARS-CoV-2 genome, identify what regions in it are highly structured, and identify very quickly small molecules that bind to those structures, which can not only disable the virus, but it can allow us to understand what are the roles of those structures in regulating, viral life cycle? What if you lock them into place? What's the consequence of that? We've also been able to deliver RNA-focused small molecule libraries broadly. We've been gratified that a lot of the chemical warehouses that supply small molecules have built RNA-focused collections off the work that we've done. And then the other thing that we've been able to do is develop cross-linking approaches that allow us to identify in cells what are the RNAs that are bound by small molecules and what are the sites of binding. And then last but not least is we've developed approaches that's the small molecule equivalent of antisense oligonucleotides, where we can basically have small molecules recognize an RNA target and bring into close proximity all the ribonucleases that cells express to trigger native quality control to eliminate specific RNAs. I want to tell you guys is that most of the data for this talk is available. The structure of the genome is on bioRxives. We have an update to this paper that was recently in the process of being put on. I want to talk about bioRxives for a second here. And so, so our initial small molecule targeting efforts has been published in ACS Central Science, but at the end of this, I'm going to talk about more of those efforts. I think one of the things that, if there's students, I think we're all students at some level. We never stop being students. But if there's younger students versus older ones like me, I guess I'd like to think I'm young. I am, and so bioRxives in the biological sciences is a relatively new invention. It's in the physical sciences. It's been used for a long time in physics. And so one of the things here that I want to stress is that the ability to have open access to data and not have to worry about it being vetted by peer review, or rather, I shouldn't say worry about, but rather delay access to that information because of peer review, and eliminating that by using bioRxives has been sort of a godsend here. And so one of the things that we've really done in SARS-CoV-2 is we've looked at not only the genome folding that we have identified and defined in a collaboration with Walter Moss, but the other thing that we've done is we sort of crowdsourced. So we've looked at other people that have worked in this area to identify regions of RNA that are structured, and then we have compared all of those studies to our studies to try to get consensus models of the entire genome. And I think that's really important. I'm an editor of JACS myself, and so I like peer review. I think peer review is really important, but I think you start to see some of the inadequacies of that in disseminating science to the consumers of it, scientists, by having that be a laborious process. Okay, so here's what we do, and I'm going to apply this pretty aggressively to SARS-CoV-2 in probably 3 slides. So the way that we look at a genome, or we look at a series of genes in a genome, we look at SARS-CoV-2, or we look at the human genome, is every RNA, a messenger RNA, can fold. Okay, and what I'm showing here is that RNAs fold and they adopt structure. And so the ways that RNA fold is they have regions of structure that are very robustly folded, and we say that they would have a high probability of structure. There are regions of an RNA that are here that I'm indicating by a single strand that are generally unfolded. And so if you're going to go and target an RNA that's unfolded, you want to use something that recognizes that RNA target by base pairing, right? Because they're openly accessible to being targeted by a complementary oligonucleotide. If, however, we want to look at and target an RNA that's highly structured, well, then we want a small molecule. The way that we go about drugging RNA is we have computational tools that I'll show you. Is we can take an RNA structure, a whole genome of structure, a messenger RNA, the entire composite of human genomic RNAs, and we can computationally determine if something has a druggable pocket. And by a druggable pocket, we mean that RNA folds into a structure, and what's shown on the top here is the 3-dimensional rendering of that 2-dimensional RNA structure. And druggable pocket means we have a 3-dimensional structure that binds to a small molecule that's shown here. that can affect its function. And now we have complementary tools that I'll show. Is that, it's nice to be able to do this and generate a hypothetical model that a certain RNA target is bound by a small molecule, but it's quite another to validate that that RNA is bound, and it's bound at the specific site that you designed a small molecule to bind. And so we've developed cross-linking experiments where you can basically take chemical cross-linkers that will enter cells in animals that are dependent on these small molecules that bind RNA, and they'll basically staple or cross-link that small molecule to an RNA target, and that'll tell us that the site in the RNA that we designed a molecule to binds. The other thing that we do, and you need to recognize here, is that binding to an RNA, much like binding to any biomolecule, is necessary but not sufficient to affect its biological function. And so in many cases, binding isn't enough to affect SARS-CoV-2 replication, infection, et cetera. And so what we do there is we can identify a druggable pocket, and we can add a second molecule onto these targets that'll recruit and activate ribonucleases that all cells express to catalytically and substoichiometrically degrade an RNA target. And I'll talk about both of these. I'm just very quickly, how we go about identifying what are the RNA folds that bind to small molecules and the small molecules that bind to RNA folds is we do a massively parallel selection-based screen. So we use evolutionary principles of selection, where we screen large RNA libraries, where these RNAs have a randomized region here where N is a mixture of A, C, G, and U, and that has 4,096 combinations. We label those RNA fold libraries and hybridize them to microarray chips in the unlabeled competitors that mimic the constant regions of the library. By hybridizing them over these microarrays or chips, where each one of these spots is a different molecule that's on a glass slide, this allows us to study a library for binding to small molecules on a surface. And then once we can identify which small molecules bind to an RNA, members of an RNA library on a surface, which is identified by having that spot on a surface have a fluorescent signal, we can go in and excise all these bound RNAs from each one of those spots. When we do that, we can identify and annotate if a given small molecule binds to an RNA with high affinity and specificity, if a given small molecule binds to many RNAs, what are the features in small molecules that bind RNAs, et cetera. And so what this allows us to do, for example, is to experimentally define affinity landscapes for small molecule RNA interactions. What's shown here is an affinity landscape for bosutinib. This is an FDA-approved medicine, and we can identify very quickly what are the RNAs that this molecule binds to. The way that we go about doing that is if these molecules have, if each RNA motif in the library has a z-obs of greater than 8, that means it's an avid binder to a specific member of this library. And so here are 18, this ligand, for example, binds avidly 18 motifs of 1,006, and those red, blue, and pink dots are biologically associated RNAs. And so what we can do is we input all of that information into what we call Inforna. So Inforna has an annotated database of all small molecule RNA fold binding interactions, and you can get access to this database, this computational software suite, on my lab webpage. Disney Laboratory. I mean, I'm not hard to find. Just don't type Walt Disney, you'll figure it out. And so what this allows us to do is take an RNA sequence, batch fold it, and then identify ligands that bind to an RNA to modulate function. And so by taking that known drug, we can identify 3 different RNA targets, microRNAs that can be bound by that ligand. So that's one way we go about studying molecular recognition. The other way we go about studying molecular recognition is we take libraries of molecules that can be FDA-approved drugs, they can be novel compounds, and we study, or fragments, and study their ability to bind RNA. And so by doing that, we've been able to annotate and identify chemical footprints in small molecule ligands that bind RNA, and have been able to build RNA-focused drug-like small molecule libraries. I will show you application of both the RNA-focused small molecule library approach to drugging an essential RNA in SARS-CoV-2, and then I will show you very quickly how we've used Inforna to go across the genome to identify lead medicines that trigger the elimination. This SARS-CoV-2 work is a collaboration in my lab between Mason, Chris, John, Hafiz, and I, and Walter, Ryan, and Jake at Iowa State. And so what Walter Moss has is a software suite where you can identify, and I'll go into it here, evolutionarily conserved structures within any RNA. And we applied that to SARS-CoV-2. So I'm going to go quickly here. This is the timeline. So you all know this, is that New Year's Eve, basically in Wuhan, China, pneumonia was reported to the WHO very quickly. 13 days after this, the genome sequence was published. I mean, this is amazing stuff, right? We started working on this very quickly, about a month and a half after that genome sequence was published. I mean, you have to understand, this genome sequence was published in January, and we have approved vaccines like a year later. I mean, this, geez, this is amazing. And it's, I'm really proud to work in a scientific community that can respond so quickly to these issues. Coronavirus as a genome is relatively small. It's 30,000 bases. It has a bunch of specific proteins that are translated in its genome, and it has a variety of RNA structures that enable for those proteins to be translated. And I'm going to talk about disabling a frame shifting element in the next few slides, and then I'm going to talk more broadly about drugging the entire genome by using Inforna and rational design. Okay, and so when the SARS-CoV-2 and other SARS genomes were published, what we did is we worked with Walter Moss's lab. And what they do is they have a program that's called ScanFold, and ScanFold allows one to scan long RNA sequences for structure. And the way this works is that you have a window of RNA sequence space, and you will fold an RNA structure by using free energy minimization. And so the way this works is you look at a segment of an RNA and you predict what is the lowest free energy or most stably folded conformation of that RNA within a window. And then you can go and look at that RNA window stability across evolutionary covariation. So you can look at all SARS-CoV-2 sequences. This allows you to identify regions of the RNA that are robustly folded, regions that are not. And I'm going to go through some of that here. And all this is published. And so this is, for example, a model of the RNA structure of a genome by using ScanFold. And so it allows you to identify regions of high structure by using this, this base pairing correlation map. Here is one thing that we found within SARS-CoV-2 is that at the time that we were doing this, Riju Das, Anna Pyle, and others were working to understand the structure of this genome. We published our work or put our work into bioarchives. And what we and others found is that a wide variety of regions of this viral genome are highly structured. Regions of high structure include the 5' UTR of this virus, which is shown here in panel B. And what you can see is that a lot of this 5' UTR is robustly structured. In fact, ways in which the virus is regulated is structured within the 5' UTR, which I'll talk about later, is that there's a RNA structure here within the start codon that is robustly folded. Another region that's robustly folded is a frame shifting element. And so, as I mentioned earlier, SARS-CoV-2, it's 30, 50,000 base pair genome. That's still not big. And so these viral genomes, in order to replicate and infect a human cell, they have to co-opt, the human's ribosome to translate the virus. And the other issue is that oftentimes these viral messenger RNAs, although they code for many, many proteins, they need to enhance their protein coding content from a single messenger RNA. One of the ways in which they do that is viruses evolve what's called a frame shifting element, which is schematically shown here. And what happens is, as a ribosome is reading and decoding a messenger RNA, a frame-shifting element acts like a clutch and a gearbox to shift the reading frame that the ribosome is translating of a given messenger RNA. And so SARS-CoV-2 has an N-1 frame-shifting element. That means that when the ribosome goes to read through this frame-shifting element, it slows it down, it gets it out of gear, hits the clutch, and it shifts it back one nucleotide. That consequence of shifting it back one nucleotide is that you completely change the protein code as you go forward through a messenger RNA. So one thing that we did is we initially focused on what was the structure of this frame-shifting element, because it has been known by a variety of experiments that if you can thermodynamically stabilize a frame-shifting element, that is, you enhance its folding, free energy of folding, is that you can basically break it. You can stop it from shifting. And these frame-shifting elements are very evolutionarily conserved, and they're evolved because it's very important to control the ratio of specific proteins that are encoded from the viral messenger RNA. And so here's a structure of a frame-shifting element. What happens is they have what's called an attenuator hairpin that slows the ribosome down. There's a slippery site and a pseudoknot part of the frame-shifting element that allows all this thing to work in concert to cause this frame shift. And so one of the things that we did is when we looked at SARS-CoV-2 is we stared at the structure of the attenuator hairpin, or one of the structured regions that slows down the ribosome to basically attenuate or facilitate frame shifting. And one thing that we recognized very quickly is that the previous assignment of this frame shifting element structure, which is shown in the middle, was inconsistent with the structure that we identified by scanfold, which is shown to the far left. Furthermore, one thing that we did is it's always good to be confident, but it's not good to be so confident that you don't look at other people's data. And so what we did is we went into bioarchives and asked ourselves, other people that had looked at these viral genomic RNA segments, what RNA structures did they identify? And their identification of these structures were done by using methods that were different than ours. And I can tell you that there are major differences between the different methods that were used to determine and study the genome structure. However, there are many sites within this genome that are consistent and conserved across the models. One of those structures is the revised structure of the SARS-CoV-2 frame-shifting element, which is shown here, which is dramatically different than the previously proposed structure. And furthermore, it is modified and different than the SARS-CoV structure. And so just why this was done is we did this by three different methods. So DMS, MapSeq, Scanfold, and RNA-Z identified these. These are the three papers that were on bioarchives. One of the papers is already published. And so given that the RNA structure was different than what was published, what was thought to be previous, and the critical function of the frame shift, the question that we had is, could we identify structure-specific ligands for this frame shifting element that very specifically bound to the revised structure and did not bind to the previous structure, to use them as not only as first a tool to understand if our revised structure is functional, because if you can disable it with a ligand that specifically binds to it, it gives you further evidence that that indeed is the structure. And also, if we can bind to this target, what would be the effect on translation of segments of the virus? And so in this case, we use these frame shifting constructs where basically we have a frame shifting element and you can measure what's the effect on translation of firefly luciferase when you have the frame shifting element upstream. And so what we did in this case is we utilized both Inforna and the RNA-focused small molecule library. And we identified from a series of molecules, about 20, I'm showing you data on 5 here that avidly bound to this RNA target and that affected frame shifting. So you can see some increased frame shifting which is shown here. And then C5 inhibits frame shifting, which is this structure. C5 binds this target with a KD of 11 nanomolar, and it has a large selectivity window for various other off targets. And so the first thing that we did is we put this into cells, and we tested it to see if it could inhibit the frame shift of SARS-CoV-2 versus SARS-CoV. It had no effect on SARS-CoV as predicted, but was able to inhibit SARS-CoV-2 frame shifting element. Now so we're inhibiting frame shifting by 25%. That is sufficient to inhibit viral replication. So you need a modest effect on frame shifting to have a dramatic effect on viral replication. And some of the work on inhibiting the frame shifting elements is ongoing. The next thing that we did is we utilized our approach to ChemClip to see if the molecules could cross-link to these RNA targets and the frame shifting elements in cells. And so the way we went about doing that is we took model cellular system that had this reporter assay in it, and we could show that we could dramatically enrich SARS-CoV-2 genomic RNA, whereas when we took SARS-CoV, we could not enrich the target. That is, this molecule, when it's mutated, will not bind the ligand. It has to have the SARS-CoV-2 hairpin. Furthermore, we can compete off the enrichment by adding larger concentration of the competitor molecule that could not cross-link to this RNA target in cells. And then in cells, we could get pretty broad and specific molecular recognition for this SARS-CoV-2 target over other RNAs that are highly expressed. The other thing that we wanted to do is equip this binding molecule with the ability to eliminate the virus and affect it, not just by inhibiting frame shifting, but by triggering the viral decay. And so the way that we go about doing this is something termed ribonuclease targeting chimeras. And what this approach is a chemical approach where we have two molecular recognition events that we can tune to facilitate degradation of RNA targets in cells. And so this is a small molecule that binds to an RNA target, is appended with a second molecule that's shown here. That second molecule will bind to and activate ribonuclease L that's expressed in cells. That will then do two things. Ribonuclease L is a monomer that is inactive. When it dimerizes, it becomes an active ribonuclease. So it activates it and turns it into a ribonuclease. And the second thing is it induces the proximity of RNA cell local to these RNA targets. It'll degrade the target. That target site will be eliminated, and then you can recycle the small molecule. Here are the structures of these ribotacs. This is the active ribotac is shown here in green, and then the inactive is in gray. When we deliver these to cells, you can see that the ribotac now is much more active. It has activity that's statistically significant, down to 20 nanomolar for affecting frame shifting and eliminating the RNA target, which we've been able to do. And then if we take these molecules that to SARS-CoV, we have no effect. If we take the molecule that can't degrade the target, we don't see any effect on any of these molecules in terms of facilitating degradation of the target. And so we're able to eliminate the target and inhibit luciferase. What we've been able to also do here is we have taken Inforna and every one of the segments that we've identified across the genome that we've been able to have small molecule ligands against, is we've been able to work with the lab of John Tajaro and show that these ribotacs can more effectively neutralize live virus in viro cells. And so, for example, these arrows, this is a binding molecule that targets a segment near the start codon. When we make that into a degrader, it gets enhanced in its activity. So it goes from an IC50 of about 20 micromolar to 5 micromolar. With one of these other segments that we can bind, the activity of the binder is about 3 micromolar, and then it can go to about 300 nanomolar. And so what we're doing now is we're in the process of looking at these studies and these systems across the entire viral genome to have a large handle on a series of molecules that can affect the virus. And one of the advantages we have, although this is very early days, I mean, we're not a vaccine that's in man, but we'd like to be able to do that, which is just absolutely amazing. is that we're able to look at these structures and inhibit viral replication in cells. And this platform that we're showing here should be readily portable across all other infectious diseases and other indications. Knowing that I'm running out of time, I want to thank my group. So this is the current group of people in the lab. Hafiz Hanif worked diligently on this. And this is the rest of my group. And I want to thank the people that funded this. I think it's pretty amazing to be in, do science and do it in a manner that allows you to work for things that, you're interested in and be able to do that broadly. And I also want to end here with thanking our COVID team, Chris, John, Mason, Hafiz, Walter, Ryan, and Jake. So thank you very much. I appreciate you guys listening to me.
Thank you, Dr. Disney. That was fascinating. Our next speaker is Dr. Stephen Thomas. Dr. Stephen Thomas is a physician scientist and a professor of medicine, microbiology, and immunology. He is the chief of the Division of Infectious Diseases and director of the Institute for Global Health and Translational Science at SUNY Upstate Medical University. Prior to joining Upstate, Dr. Thomas spent 20 years in the U.S. Army Medical Corps, serving at the Walter Reed Army Institute of Research and completing his career as the Institute's Deputy Commander for Operations. Dr. Thomas specializes in the study of viruses and vaccine development. He played key leadership roles in the U.S. government's response to the West Africa Ebola outbreak and the MERS and Zika epidemics, and was instrumental in the development and advancement of vaccines for each virus. His current research activities include developing vaccines and drugs against mosquito and tick-borne illnesses, vaccines against opioid use disorders, and field studies exploring global health challenges. During COVID, Dr. Thomas led SUNY Upstate's Incident Command as they prepared and responded to infections in central New York. His team initiated numerous experimental COVID treatment trials, and he is the coordinating principal investigator for Pfizer's COVID vaccine trial. Dr. Thomas advises the World Health Organization, the U.S. government, and academic and industry partners on global health and biomedical research. He is a frequent contributor to the Forbes magazine and the scientific literature. Dr. Thomas earned his Bachelor of Arts with honors in biomedical ethics from Brown University, his medical degree from Albany Medical College, and completed his internal medicine residency and infectious disease fellowship at Walter Reed Army Medical Center. Dr. Thomas, let's just take a minute to make sure your slides are running. And they are. Dr. Thomas, please feel free to begin.
Thank you very much. Can you hear me okay?
Yes.
Thank you. Well, I'd first like to thank Dr. Disney. That was a great presentation, and you taught this old student a lot with that, so thank you very much. I'm going to talk about COVID-19 vaccines today. Here's some disclosures. As was mentioned, I'm an academic, but I'm one of the principal investigators on the Pfizer trial, and I supported them in their preparation for their FDA meeting. So we'll get right into it. So this is how SARS-CoV-2 infects cells. So we have the spike protein, which is the business end of the virus, and it's sort of the key that goes into the host cell. So cells could be in our nose, could be in our lungs, or other organs, and the lock, which is the ACE2 receptor. So you can see that the virus spike protein recognizes the receptor. It binds to that receptor. There's conformational changes. The virus gets brought through cell fusion into the cell. And as was mentioned, it undergoes a replication process. Baby viruses emerge from the cell, and they go on to infect other host cells. So that spike protein is incredibly important to viral replication. And as we found out, and as we've known, there are numerous organ systems that have these ACE2 receptors. So they're not just within the respiratory tract or in the nasal passages or in the lung, but you can find them in the GI tract. You can find them in the genitourinary tract, so the kidneys. You can find them in the nervous system, so in the brain, the liver, and the heart. And as a result, COVID, the disease caused by SARS-CoV-2, really is a systemic multi-organ disease. And if you recall, when we first started to talk about, and the WHO and the CDC started to advise people on what symptoms of COVID might look like, you can remember initially it was We think this is a pneumonia. So if you have fever, shortness of breath, and cough, that could be COVID. And then it expanded to include nausea, vomiting, abdominal pain, diarrhea. And then it expanded again to loss of taste or loss of smell. And then it expanded even more to, you may remember COVID toes, right? So these lesions that we have on our skin. And, for those of us that take care of COVID patients, this slide will speak to you, because we've seen numerous case reports and case series of, young people having stroke and people losing a sense of taste and losing a smell. People who have acute kidney injuries end up on dialysis. People that have elevated liver enzymes showing that they have inflammation within their liver, nausea, vomiting, diarrhea. People that, because their gastrointestinal tract is affected, they have translocation of bacteria that should remain in the gut, and it gets into the blood, and that can cause sepsis and lots of other problems. People that end up having these, they have depressed cardiac function, or they go into a shock because the pump just isn't working, or they can sometimes even develop these fatal arrhythmias. I remember when we were speaking, and early on in the U.S. epidemic, we had a conference call on a Saturday with 3 different hospitals in Wuhan, China, and the clinicians were telling us how they would have people that would survive the intensive care unit only to make it to the medical surgical floor and die of a fatal arrhythmia. And then, of course, the development of clot, whether it be clot in the lung or clot in the heart or in the venous system. So this really is a multi-organ and systemic infection, and we believe it tracks with the concept that the ACE2 receptors are all over our body, and that's how the virus attaches and gets into cells. So knowing that, it would make sense that a rational immunologic target for a vaccine or a therapeutic, for that matter, would be the spike protein. So the spike protein binds to ACE2 receptors. That's what gets the virus to fuse to the cell. That's what allows the genetic material to get into the cell, new viruses to be made and infect other cells and other organs. So the goal would be that if you could develop an immune response, and when I say immune response, I'm thinking very comprehensively and broadly, but I'm just mentioning antibodies here, as an example. But if we could develop antibodies to this spike protein, then we may have the possibility of blocking or reducing infection, and therefore preventing or reducing disease, either the occurrence of disease or the severity of disease. So the goals of vaccination, so the highest bar there would be to prevent infection, reduce the total number of infections that are occurring. The second would be to reduce, even if someone was to be infected, you make them unable to be infectious. So you try to use vaccination to reduce transmission from person to person. But really, the 2 goals that that are kind of the near-term targets would be reduce clinical illness, right? So people who get vaccinated, even if they get infected, they have an asymptomatic, or they have a clinical illness that is so mild that it does not require them to access the healthcare system. Because if we think about what the global public health burden is of COVID, it's that you're taking a susceptible, non-immune global population, you're introducing a virus into that population, even if the case fatality rates are 1.5%, 2%, which does not seem like a lot. There are going to be so many people that are accessing the healthcare system that, especially in the United States, where we operate at about a 90% capacity to begin with, you're going to quickly overrun that healthcare system. And then, of course, as we've all seen and we're living and experiencing, the economy and business and education and overall quality of life is, of course, impacted. And so the hope would be that through mass vaccination, we might be able to impact some of these things. Again, the clinical outcomes piece is really important, and I'm not just saying that because I'm a clinician. But if we know that if the majority, let's say about 30 to 50% of people who get infected have an asymptomatic infection, but they remain infectious, right? They can continue to infect other people. Of those people who become clinically ill, we still believe about 80% of them are going to be, mild to moderate illness. They may need to go to the doctor to get diagnosed, or they may need to go to the doctor to ensure that they're stable and get symptomatic care. But then there's this severe and critical portion, which is about 20% of all the people who end up getting sick. And it's those people that need to come into the hospital or receive infusions or end up in the intensive care unit or on a ventilator and ultimately may die. And so whether you have a severe case and you're accessing the healthcare system, or you have a critical case and you're accessing the healthcare system, you are still creating a huge burden on the resources. And there's also these secondary costs. So, I know in our hospital, so we had an initial blip in about April or May, and then the wheels started. We were doing great during the summer, and then the wheels started coming off like around Labor Day, and then Halloween, and then Thanksgiving, and then the December holiday season, where people were just gathering in large groups, and we got so overwhelmed. Just in my little county here in central New York, we were having more than 450 cases a day. The hospital was completely full. We could no longer do elective surgical procedures. We had to shut down. And these are people that have real medical problems that need surgery, to include certain malignancies and cancers that they needed to have surgery. We had no space. We had no beds. We had we had decrease in staff because staff were getting infected out in the community. And so it delayed treatment. So there's all these secondary and tertiary costs of COVID. So the idea is, if through vaccination, if you can transition people who would, without vaccine, be severe or in the critical category, if you can transition them to a mild or moderate, or the best case would be asymptomatic category, again, you can help to preserve that healthcare system. So here is the lay of the land right now. I saw this with Ebola, we saw it with Zika, saw it with MERS as well. You get a lot of horses that start the race, very few of them end up finishing. One thing what we've seen with COVID is there's been some real staying power in terms of the numbers of vaccine candidates that have been put into different phases of development. This is looking primarily at clinical phase. So there are other vaccines that are in preclinical phase that are not listed here, but it's a pretty good number. It's about 100 or so candidates that are still out there going through the clinical development process. There are lots of different vaccine approaches. What I've tried to do is put green boxes around the pros and put red boxes around the cons. So let's just look in the middle first. So you take live attenuated virus vaccines, or killed vaccines, and activated vaccines. These are, and even subunits. So these are classic vaccine approaches that, if you look at the green boxes down here, they have proof of concept. They have been used before. They have been licensed because they've been demonstrated to be safe and effective in preventing these other diseases. And so that's an advantage of these. They also are known to develop immune responses that we are familiar with characterizing. And so in the example of live attenuated vaccines, they have a nice comprehensive immune response. So antibody development, cell-mediated immunity, it can last for a very long time. Yellow fever, for example, one shot, you're immune for the rest of your life. The problem is live virus vaccines are not good for people with compromised immune systems, and we typically don't vaccinate pregnant women with live virus vaccines. Killed vaccines, sometimes the immunity doesn't last as long. Sometimes you need to add adjuvants to support their immunogenicity. And with SARS and MERS specifically, so with the other two severe coronaviruses, there was animal data that demonstrated that people who got vaccinated and then subsequently exposed to a wild-type virus, that they might actually have a worse clinical outcome, worse pathology than those that had not been vaccinated. And people still remember the inactivated RSV vaccines that caused more severe disease and killed some children. So that's something that manufacturers would have to get around. So then we have viral vectored vaccines. So this is your Oxford AstraZeneca vaccine, your Janssen or your J&J vaccine. And then the DNA an RNA vaccine. So although Johnson & Johnson has licensed an Ebola vaccine, most of the other viral vector vaccines have really just been in kind of veterinary medicine. So there's a little bit of a regulatory obstacle here that you might have to overcome. And the same was true, of course, for DNA and RNA vaccines. They had never been licensed before for human use. So this is kind of the landscape. But regardless of which approach you take, whether it's a viral vectored approach, a DNA vaccine, an RNA, live virus, protein-based, it's all a common pathway at the end, which is, again, back to that, the business end of the virus, which is that spike protein, and the idea that if you can make an immune response against that spike protein, you have the opportunity to reduce the burden of infection and therefore reduce clinical illness. So I'm going to focus on the messenger RNA vaccines because those are the ones that are currently under emergency use authorization in the United States. So how does it work? Well, you have the spike protein and you have the messenger RNA sequence that codes for that spike protein, the directions that the cell will use to make spike protein. You code it in this lipid coating and you inject it into the muscle. It gets into the cytoplasm of the cell. The cell starts to read the messenger RNA, starts making spike proteins. Parts of the spike protein get expressed on the cell, or they go out of the cell, and the body recognizes them as foreign, and they start to make a mount an immune response against that spike protein. So you now have built your army, such that the next time you are exposed, let's say you're in a room with somebody who's infected and they cough or they talk to you and you don't have a mask on and you get infected, the body is going to have a very robust adaptive immune response and a very quick memory immune response and neutralize those virus particles that that you ingested in that encounter. So that's kind of the theory behind how these messenger RNA vaccines work. Vaccine development. One of the things that as a vaccine developer, it's interesting to see how many people are now interested in vaccines and how they're developed compared to before COVID. Historically, it used to take up to 10 years to develop a vaccine, well over $1 billion US, and it was a very slow process, and the likelihood of success was quite low. If you had 100 candidates enter in the pipeline, you would be lucky if one of them received a license and a marketing authorization. And the reason, and this is what people expect, they expect this long, drawn out, very, very expensive kind of plotting process. We've been talking in the field for, 5, maybe even 10 years that it really doesn't have to be this long. Well, what were some of the reasons why? Well, first of all, it is a low likelihood of success and a very high price tag, right? Over $1 billion US. And so the companies and the developers are going to really try to mitigate financial risk. And as a result, they take very slow, sequential, pragmatic approaches to developing vaccines, ensuring that every phase, going from your discovery and your preclinical animal phase, you need to have very, very robust and very convincing data to go to the next phase, which is to do some small-scale clinical biomanufacturing and enter into your small safety studies, dozens of people. And then you're going to look at that data, and then you're going to go to the second phase, which is also concerned about safety, but also looking more at immunogenicity, looking at dose and schedule and series and things of that nature. And then if that data is highly compelling, that's when you're going to write the big checks. You're going to do your big commercial biomanufacturing scale up and do your huge phase 3, what we call clinical endpoint study. So prove that the vaccine has some kind of clinical benefit. And if you're lucky to get through all of that, you file your biologic license application, your BLA. Hopefully the FDA gives you the thumbs up, and then you can launch your product commercially. Well, again, you can imagine how all those touch points and all those reviews and all that can take a really long time. It also took a really long time historically for regulators to communicate with sponsors. Now that got fixed, and as of recently, even in non-public health emergency times, their turnaround time for review of a final packet was about 10 months, which is better than the 2 years that it used to be. Another big element in terms of why it typically takes so long is you need to demonstrate efficacy. You need to demonstrate that there's a clinical benefit. If you work on a disease where the clinical attack rate, so not the infection attack rate, not what percent of people in a given space are going to get infected, but what percent of people are going to actually get sick so that you can measure the signal. If that occurs at a very low rate, less than 1%, for example, you're going to have to do big, huge studies, and you're going to have to follow people for a really long time so that you can collect enough cases to show that there's a difference between the vaccine group and the placebo group. But what happened with COVID? So first of all, a lot of people, not on this call, but a lot of people in the community were baffled how this brand new technology could just come on the scene and solve a problem. Well, we've been working on messenger. We as a scientific field have been working on messenger RNA technology for over 30 years, right? And they had, for the last 10 years or so, 5 to 10 years, there have been numerous clinical trials with messenger RNA-based vaccines, whether it's for Ebola or flu or Zika, HIV, for example. And so there was already a lot of technology that had already occurred. They weren't starting from ground zero. The first is that there were a lot of parallel processes, so especially with manufacturing. And this is where a lot of money was put up at risk. In the case of Operation Warp Speed, the companies that belong to Operation Warp Speed, that $10+ billion was underwritten by the US taxpayer. In the case of Pfizer, they decided to underwrite it themselves. If you look at both the R&D and look at the distribution work that they've had to do, it's probably close to $3 billion. So because of the pandemic, people were willing to take that financial risk and produce at commercial scale large batches of vaccine that it's possible they may not have worked. They may not have been demonstrated to be safe and effective, in which case they would have spent a lot of money for a failed product. The other thing is that there was engaged regulators. So as you can imagine, lots of singular focus, head start in technology, everybody keeping their eye on the prize, and the FDA having very specific discussions with regulators and explaining in very clear terms what the expectations are. It kept people on a straight path, and so it allowed folks to move very quickly from generating that initial first in human data, immunogenicity data, going right to phase 3. And then, unfortunately, but good for the vaccines, unfortunately, there was so much infection going on, so much transmission, and so many people getting sick in the 150, in the case of Pfizer, the 150 trial sites around the world, that they very quickly were able to collect the number of cases that were needed to do their interim analyses and support a request for emergency use authorization. So that's kind of the difference between kind of the classic vaccine development phases and what we saw with COVID and why. So now I just want to give you sort of a side-to-side view of the two vaccines that are under emergency use authorization. Both were published in the New England Journal. We published ours, I think, a week or two before the Moderna vaccine. And I just want to kind of give you the side-by-side. So Moderna's on top, Pfizer's on the bottom, and basically very, very similar. So about 30,000 people in the Moderna trial, about 38,000 people in the Pfizer trial at the time that this data was published. Moderna, you get the first dose on day 1, second dose on day 29. With Pfizer, it's day 1 and day 21. The trials were kind of similar. So we did not actively enroll anybody who had history of a COVID diagnosis or had a known antibody positive status. So that was done through medical history. But on the day you got both your vaccines or both your injections, we were all blinded. We didn't know who was getting what. We would do a swab, and then we would draw a blood sample. You get your 2 doses, we follow you for safety, and then both of these studies are about 2 years long. And so we're going to be looking at sequential visits over time to look at things like durability, et cetera. So very similar studies. When we think about safety in vaccines, we think about local safety and we think about systemic safety. Local safety is where were you injected, right? Everyone's mostly injected in the deltoid. So we look at pain, redness, swelling at the point of injection. So let's look at the top. This is Moderna. What you're looking at is the first two bars represent placebo. The second two bars represent vaccine. Dose 1 first, dose 2 second. The colors represent different grade severities. So grade 1 is, for example, with pain, my arm hurts, I know it hurts, but I can do what I need to do, not a problem. Grade 2 would be, my arm hurts and it's interfering a little bit. I'm not going to be able to exercise today, for example. Grade 3 would be, my arm hurts and it is preventing me from doing what I want to do. I can't use my arm today. So what you see is locally with Moderna, about 75+% of people have some pain, but those people predominantly say it's mild. And then there's a smaller group of people that say it's moderate. Very few people say that it is severe. So that's good. There's very little redness, very little swelling, and dose 2 is a little bit higher than dose 1. Similar sort of observations with the Pfizer vaccine. So Pfizer split into their age groups, so 16 to 55 and greater than 55. And again, what you see is pain is the predominant symptom. It's anywhere from 7 to 8 out of 10 people. It's pretty equivalent between dose 1 and dose 2, and it's mostly mild to moderate. In terms of resolution, Pfizer, it seems that based on the reports that it resolves a little bit quicker. So 1 to 2 days after the symptoms start, Moderna a little bit longer, on average 4 to 5 days. And just speaking personally, that was the symptom that I had predominantly, and it was after my second dose, pretty significant arm pain, but it lasted about 24 hours and then went away. Now, in terms of systemic side effects, so we're going to look at Pfizer first. What you're looking at here is, again, on the top is the younger age group, 16 to 55. On the bottom is the greater than 55. Dose 1 is first, dose 2 is second. Same color scheme, so mild and moderate is green and blue. What you see is we get about 10 to 15% of people have fever after their second vaccination. Very little after the first. It's mostly after the second. The other thing that we see is fatigue and headache. So that people are reporting fatigue and headache mostly after the second dose, well, more after the second dose than compared to the first dose. And it's about 4 to 5 to 6 out of 10 people. What's interesting is the older people, so those greater than 55, they have less reactions than the younger people do. And these, just so you know, these are all graded by the volunteers. These are not graded by me. The other thing you see here is we have the placebo group. So I think that's important. So you have 47% of people after the first dose complaining of fatigue in the vaccine group, but you also have 33% of people in the placebo group who are complaining of fatigue. If you look at headache, here you have 52% of people after dose 2 complaining of headache, but 24% of people who got salt water also complained of headache. So this is why we do these blinded placebo-controlled trials. So overall, fatigue and headache were the most common symptoms. You do have some fever after dose 2, about 1 out of 10 people. Dose 2 symptoms, worse than dose 1 symptoms. Younger people with more symptoms compared to older, but everybody says mild to moderate, begins within 1 to 2 days, and the symptoms resolve within 1 to 2 days. Moderna, similar. So fever after dose 2, about 15% of people, mostly mild to moderate. Headache and fatigue and muscle aches, again, more after dose 2 than after dose 1, mostly mild to moderate. Although we do see, if you look at the red bars, we do see an increase in the overall number of grade 3s after dose 2, but still, minor compared to the other grades. And the symptoms last on average 2 to 3 days. So here's efficacy. So if we look at Moderna's on the left, Pfizer's on the right, these are cumulative incidence or cumulative event curves. Remember, Moderna vaccine, day 1, day 29, those are your 2 doses. Pfizer, day 1, day 21, or day 0, day 21. The good thing with both of these vaccines is that you see it's the gray line is placebo, the blue line over here is placebo. What you see is that very early, even after the first dose, these cumulative incidence curves start to separate. And it is very clear they are not going back together. And if you look at the intention to treat, meaning what was the efficacy after a single dose. It's about 93% with Moderna, 82% with Pfizer. And if you look at overall efficacy, meaning the per protocol efficacy, so what was the efficacy when you start counting cases 2 weeks after your second Moderna dose or 1 week after your second Pfizer dose? Very similar, 94 to 95%. What you can see here for both vaccines, but here it's blown up so it's a little bit easier to see. After a single dose of Pfizer vaccine, around day 10, 11, 12, those curves start to separate. And this shows that there is efficacy after a single dose. And they calculate that efficacy of about 50%. The issue, and this is sort of the basis for why some areas like the UK, for example, have said, you know what, it is okay to delay your second dose because there's efficacy after the first dose, and we need to maximize people getting, the first dose in. I don't agree with that, but this is sort of some of the data they're looking at. The reason I don't agree with it, and the problem is, there was in both these trials, such a high rate of compliance with the vaccination schedule that we really don't have a significant number of people who only got one dose to judge whether or not this would have been enduring or whether or not these two curves would have come back as, let's say, antibodies waned immediately after the first dose. So high level efficacy after 2 doses, partial efficacy after a single dose, at least until dose 2, and then the efficacy appears to persist after 2 doses. If you look at subgroups, the bottom line here is that with Moderna, it looked like the younger people had a higher efficacy than the older folks, 95% versus 86%. But the numbers are really small, and we need to see what the complete database looks like after a more prolonged period of time. There did not seem to be any difference in efficacy across medical comorbidities that people had, or again, based on race or ethnic group. But again, the numbers of infections, once you start kind of picking out subgroups out of the total group, you start slicing the pie, it gets very small, and it's very difficult to conclude with a lot of confidence. But the trends are certainly supportive, and the same is here. So the trends support that older and younger people have a similar efficacy when they get the Pfizer vaccine. Males and females are similar, and there's equivalence across race and ethnicity, with the caveat that there are low case counts, at least at this first reporting, but we anticipate we'll have more cases to maybe feel a little more comfortable about making a definitive statement. Severe disease, there was only 1 case of severe disease in the Pfizer vaccine group, none in the Moderna vaccine group. Again, very small numbers, but there's at least a trend that, in fact, the vaccines may have a high level of efficacy against severe disease. This was an endpoint that was put in partly because of what I mentioned with inactivated vaccines, because with other severe coronavirus vaccine candidates in the past, they saw potential for, I don't like to use the term enhanced disease because it implies an immunopathologic mechanism that we don't know if that's necessarily the case. But when we saw severe disease in animals who were vaccinated compared to those who were not, people were concerned that vaccines could actually make it worse, which is why they wanted them to check for severe disease. If there was equivalence between the vaccine and placebo group, or if there were more severe cases in the vaccine group, we'd be in a tough spot right now. But fortunately, the trends look positive. So where are we? Well, it looks like 28 million first doses have been shipped out in the United States, which cover about 8.5% of our population, and the tally is about 50% of those first doses have been used so far, which represents about 4.3% of the U.S. population. I check these numbers frequently, and the percent of people being vaccinated based on the denominator of how many first doses have been shipped, that number continues to rise, which is a good thing. But clearly, we still have some issues we got to tackle with that. There was this concern about adverse events. So, for example, allergic reactions. The first report by the CDC and the MMWR basically showed that it would about 11 cases of anaphylaxis per million doses. This was based on about 1.9 million doses of Pfizer vaccine. And the majority of these occurred in women. The majority of these reactions occurred within the first 15 minutes. And the vast majority of people that had these reactions had a documented history of allergies or allergic reactions, to include people that had had anaphylaxis to other vaccines. Why is this happening? Well, people aren't totally sure because the vaccine ingredients are pretty streamlined, but there is this polyethylene glycol in both of the vaccines. You can also find it in shampoo and other ingredients in certain medications. But this has been known to cause allergic reactions in people, and so they're investigating it. I haven't really seen too many other hypotheses out there. As I mentioned, variations in the dose or the schedule. I don't, particularly think it's a good idea right now in the United States. I'll just speak for the U.S. The FDA doesn't either, and so as it stands right now, we're going to keep dosing the way the trials demonstrated them to be safe and effective. Mutations matter. Mutations matter because obviously they can impact the protein. They can impact the shape of the protein. They can impact epitopes, and this might change how our diagnostics work. It might impact our therapeutics that we're using, and of course, everyone's concerned that it could cause escape mutants. And so, you induce immunity in somebody with a vaccine. They get exposed to a variant or a mutant, which that immune response no longer works against that mutant, and that would be a problem. So, this isn't the first time we've seen a variant. We've had one variant already, this D to G variant that came out of China that, basically was replaced, the G strain replaced the D strain globally back in May. They did in vitro experiments that demonstrated that the mutation was in on the spike protein, and that it was that it showed in vitro that it could make the virus more likely to infect a cell. And so that was concerning to people, but there wasn't too much about this because for the most part, if you work in viral vaccines or you study viruses, mutations are just kind of the way it is. It happens. But then we started to see some of these new lineages and these new strains, one coming out of South Africa with some very concerning mutations in the receptor binding domain of that spike protein. If you look at the yellow here, you see that in a very quick amount of time, that variant replaced the other variants that were and other lineages that were being collected in South Africa. Shortly thereafter, the UK then started to find their own B1.1.7 variant, and quickly you can see how the percentage of variants of concern replaced the strains that had already been circulating in that area. And you can see here this heat map of of the UK, or at least England, and how the variant very quickly became the predominant variant. When they did some modeling, they came up with 4 different scenarios that might explain the epidemiology. You're looking and one was, well, there actually is increased transmissibility of this variant, or they thought, well, could it be an immune escape? Could it be increased susceptibility among children? Could there be a shortened viral replication time? The purple represents their model. The black represents the actual data. And you see up at the top here, the increased transmissibility hypothesis at least models most closely with the data. So this is the main theory right now. Japan has identified a new variant. California has identified a new variant. So what have people done? So this was provided to the FDA. These are variants of the receptor binding domain in the receptor binding domain for 18 different pseudoviruses that were created to look at whether or not neutralization by vaccine-induced antibodies would persist across these variants. And as you see, there's not much variation. There's effective neutralization across these pseudoviruses. There have been some papers that have recently been posted. And again, what you're seeing here is you're seeing sera collected from vaccine volunteers and we're looking at neutralization between what you could call maybe the old strain or the wild type and this new variant. This particular group just looked at one variant, so this N501Y. There was equivalence in neutralization capability. This group actually looked at the Wuhan reference strain compared to the UK 1.1.7, and again, you can see there's consistency in neutralization, but the story is not all good. I literally just added this before this. I just saw this paper today. Just came out, I think, 2 days ago. And this looked at 17 different monoclonal antibodies with different mutations in them. And for 3 of them, there was a statistically significant decline in neutralizing capability based on both Moderna and Pfizer-induced neutralizing antibodies. Now, very, very small numbers, I can tell you that, but it's at least concerning enough, or it at least wants to, should motivate us to continue to look at this. I think in the end, this has to translate. The lab findings somehow have to translate to biology, and biology is going to be looking at people with breakthrough and looking at vaccine recipients who have breakthrough infections, collecting their viruses, and sequencing their viruses. So remaining questions. Obviously, we want to know how long are these vaccines going to work? How long will they protect us? We want to understand, do they actually prevent infection? Can they make us non-transmissible, or is it just a clinical endpoint? Are we going to, if these variants continue, are we going to need to make new strains? Is this going to be like flu, where we need a new vaccine every year? What about other populations who are at risk that were not included in the trial? So the very young, the immunosuppressed, women who are pregnant. Vaccines don't save lives. Vaccination is what saves lives. And about 50% of Americans say they're not going to get vaccinated. I don't know if that's changed in the last couple of weeks with vaccine rollout and people who are getting vaccinated serving as examples that it's safe. But we really have to address this hesitancy issue. And then, how long is it going to take to achieve vaccine-induced herd immunity? At the current pace, it's going to take a really, really long time. But is that something to shoot for? I think it is, because we've done it before. And then, of course, it would be great if we could define either a mechanistic or non-mechanistic correlate of protection to speed up either development of second-generation vaccines, because we need as many as we can get, or could it, help us with annual, if we need to, change the vaccine strains annually or every couple of years, that could certainly help us to, meet regulatory requirements. So thank you very much for your attention. I appreciate it. I will stop sharing and pass it back over to our hosts.
Thank you, Dr. Thomas. That was interesting. Now, in the time we have left, let's get to our listeners' questions. And I'll start with you, Dr. Thomas, because I think you touched it there at the end of your slide. Do you foresee the current vaccine as being sort of a seasonal thing, sort of like what we do for the flu?
I certainly hope not. The classic or community-acquired coronaviruses, so there's 4 human coronaviruses that cause about 15 to 30% of the upper respiratory infections we see every fall and winter. The immunity we develop to those, it seems to be pretty fleeting, and that people can get infected year after year. But that's different than vaccine-induced immunity. So I am hoping that the immunity that we see with these vaccines is going to be a little bit more enduring than what we've seen with the community-acquired coronaviruses. But this is why these trials are multiple years, and this is and I know that groups are prospectively looking at this over the long term. I will say though that if, at least with the messenger RNA vaccine platforms, if there is a need, these are good platforms because you can create the new candidate within a matter of weeks, through your plug and play, and manufacturing can occur with great speed. So we'll see, but I hope not.
Yeah, thank you. Dr. Disney, our next question is for you. what are the advantages of small molecules versus protein-based approaches?
Right. Well, okay, protein. So if the protein-based approaches are the vaccines, right? So, not everybody is going to be vaccinated. And so there are going to be people that are suffering from issues that have to be treated. If you're talking about delivering antibodies for people that have COVID, what's the advantage of, say, an antibody treatment versus a small molecule?
Yeah.
Well, you can manufacture small molecules in principle much more quickly than you can some of these biologics. And so I think that's a major advantage of it. Yep.
Okay, thank you. Dr. Thomas, how does the vaccine work? Does it protect you from getting the virus, or does it help you launch an immune response?
Yeah, the only thing, well, it works through generating, excuse me, an immune response against the spike protein. The only thing that we can definitively say right now is that it reduces your risk of developing COVID, so developing disease, by 94 or 95%, depending upon which vaccine that you get. The clinical endpoint that we measured was any COVID symptom plus a positive PCR that was a case, right? So that's kind of where we're starting. So the only thing we can say is that the vaccines protect you from developing COVID disease. Now, do we know whether it prevents people from becoming transmissible to other people? Do we know if it achieves the highest bar, which is to prevent infection? We do not know. The samples were collected and the data were collected that might give an indication, but those are study designs that are a little different than how these efficacy trials were done. But that's under active exploration. And boy, it would be great if this was a transmission blocking vaccine as well as a clinical disease attenuating vaccine.
Indeed. Dr. Disney, with the evolution of SARS-CoV-2, I think there was a recent evolution or a new strain. Do you see the frame shift portion of SARS-CoV-2 being as important even in new iterations of the virus?
Yeah, so one of the advantages of this approach is you can very quickly identify if the RNA structure is mutated. I do not believe that frame shifting element is mutated. In fact, I'm certain it isn't, and that's generally conserved. I would say one of the other advantages with the Inforna approach is that if, for example, a small molecule were to evolve resistance, it would have to change its structure by only having 3 other nucleotides to put into it while still having the same function and not binding your drug. And so one can imagine a scenario where resistance to medicines that target RNA structures that are important in function could be harder than evolving resistance to molecules that target a protein, which has 19 other amino acids to substitute. So, yep.
Thank you, Dr. Disney. Dr. Thomas, will the Pfizer approach of a trimeric receptor binding domain as opposed to a full-length spike make it more susceptible to mutations, do you think? Or is it too early to say?
Yeah, I think that's a good question, and I think it gets to the concept of quantity versus quality, right? So these, the spike protein has a number of different epitopes on it, and some of them would be some of them are possibly more important than others, and so the idea would be that, well, if I have one really highly avid binding antibody for a specific epitope that prevents some major conformational change, for example, then or prevents it, it is like the one that will prevent it from binding, then in that case, it's a quality issue. But in the event that maybe you produce antibodies that don't go after that particular epitope, but you have a variety of antibodies that attach to other epitopes that, quantitatively can take out the virus, then that's another issue. So they are starting to matter of fact, I think the last paper that I referenced gets into the breadth of epitopes and binding locations that these vaccine-induced antibodies attach to. But that's the kind of information that we need to explore, because I think as the question implies, how at risk are we from these variants? And so, but that's I think we don't know yet, but that's kind of my initial thinking: quantity versus quality, and let's see how it turns out.
Thank you, Dr. Thomas. Dr. Disney, how long does it take to build one of those small molecule libraries and to test it?
Yeah, so to build the small molecule library, you can actually get them from commercial suppliers. So you can basically order an RNA-focused small molecule library now, and you can do the testing for it to bind to specific RNAs, at least in a high throughput mode, in a day or so, and then you have to confirm that's binding. So the testing for binding can go very rapidly, and then on top of that, you have to test it for affecting viral translation, or the viral lifetime. So all in all, right, you can go from an idea to a lead molecule in a month or so if you're on the ball with this infrastructure.
Oh, that's pretty cool. Thank you, Dr. Disney. Dr. Thomas, why do you think there were more side effects after the second dose?
Yeah, I think the side effects are, and this is not just with this vaccine, but we see oftentimes reactogenicity tracking or being associated with immunogenicity. So this is what we call a homologous prime boost, right? So both vaccines are the same, so that's homologous. The first vaccine primes the immune system. The second vaccination boosts the immune system. And so what I think we're seeing here is we're seeing big, huge boosting immune responses after the second vaccination, and that is and the way that happens, right? The way that antigens are captured and processed and presented to the immune system, it causes cytokines and other things to happen, and I think that's why people have fatigue, and they can have headache, and they can have in some cases they can have fever. So I think it's and that may be the reason why older people seem to have less symptoms than younger people. I don't think it's because they might not complain as much. I think that they might have some level of, a very young person's immune system is different than, a middle-aged person's immune system, different than an older person's immune system. And so I think there's probably some, immunosenescence there with the older folks, and they just don't have that level of robustness that makes them, have the reactions that the younger crowd does.
Thank you, Dr. Thomas. Another question for you. Why do you think we haven't turned to mRNA vaccines sooner? Why do you think it's sort of coming of age during the pandemic?
Yeah, as I mentioned, they've been exploring these for a really long time. And I would encourage people, if you get a chance to read about, how they were developed. Again, this was being developed for about 30 years, and there was a lot of no thank yous from granting organizations and funders and federal funders early on. I think that there's, it represents vaccine development is high risk to begin with, and any kind of new technology that has not been in front of a regulator before, I think is considered high risk. I do know there were a lot of big pharma that have been looking at this technology for a while, but previous things that they've worked on, like Ebola and Zika, it was very, very difficult for vaccines to meet that efficacy endpoint that I was mentioning, because the epidemiology was not in line with the development life cycle. So by the time people were ready to go to the field, the diseases had already started to go away, and they just weren't going to get they weren't going to get their chance. So, I think those are some of the reasons. But this is, it's a big deal to get, in my opinion, it's a big deal to get a new class of vaccine, licensed, even if it's not licensed, but even just to get an EUA. I think it's a really big deal to get a new class with this type of data because of the plug and play possibilities.
Thank you. And now we have time for just one more question, and I'm actually going to direct it to both of you. I'll start with you, Dr. Disney, first. What is the next biggest step you see for small molecule research, specifically as pertains to SARS-CoV-2? And Dr. Thomas, I'll ask you to follow up for vaccines just after. Dr. Disney?
Oh, I think for the whole area, not just for SARS-CoV-2, is I think there's a lot of excitement in small molecules that target RNA, not just to position it for SARS-CoV-2, but a host of genetically defined diseases where there's no known cure. And so, frankly, it's progressing more molecules into the clinic. I think this is a new modality like the mRNA vaccines were, and the quicker you can get them in demand and show that they're safe and efficacious, the more it's going to accelerate the whole field. So it's not, in some sense, it's a new modality, and the challenges are similar for each one of them as they come up and start to be developed. So it's got to fulfill its promise, and the only way to do that is to have demand.
Thank you. Dr. Thomas, what do you see as the next big thing that needs to be answered for vaccines? It could be for SARS-CoV-2 or at large.
Right. Yeah. No, so I think, first of all, the vaccine development process, I think people are now believers that it really does not have to take the 5 to 10 years that it has traditionally taken. The discovery process is going to be what it is, right? It's going to take a long time to think up prototypes and to do that bench work and preclinical work. But I think once it's ready to transition to people, we've seen and shown that it can move much faster and be very safe as it does. That's the first thing. I already mentioned that I think a new class of vaccine platform is a great thing for SARS-CoV-2 specifically. So, if you get an emergency use authorization, you have a requirement to pursue a full license on a reasonable time frame, or the FDA can take away your EUA. And so, and I think the first time point is 6 months of safety and efficacy data, which will come this spring. So I think that's the first thing. We're going to see both of these messenger RNA vaccines present new data to the FDA at some point in the spring. And there's other vaccines out there. So, we're not sure what the AstraZeneca Oxford group, they had about 70% efficacy. We're not sure if they're going to pursue licensure in the US or not, but that's, they've been through a very large study. Novavax has started their phase 3 in the United States. We still have Sanofi, who's in the game as well. And so, there's a lot of other vaccine candidates that are out there still kind of percolating along. But, listen, we have 7.5 billion people on the planet. None of us were immune to SARS-CoV-2 as of a year ago. So, as many vaccines as we can get out there, the better.
Great. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the DDN website, and you will receive an email notifying you when the webinar is available on demand. On behalf of DDN, I would like to thank our speakers, Dr. Matthew Disney and Dr. Stephen Thomas, as well as our sponsors, ACD Bio, Taconic Biosciences, Cayman Chemical, and Olink. And of course, our thanks to you for listening. Have a great rest of your day, and goodbye.